My pleasure to introduce Brian Walker here today. So Brian first started his work in marine mammals, and he's seen the light and come across to penguins, which is fantastic. So he started his master's on harbour seals in Dalhousie University in Canada. He then did work with looking at seals in the Antarctic Peninsula for 4 years, and then he came across to do not came across here, but he went to Washington University to do work on Magellanic penguins, looking specifically at the impact of tourism on the physiological response of penguins. And now, like everyone, we are really increasingly worried about plastic pollution in our oceans. So Brian is doing some extensive work on microplastics in penguin feces and other seabird species at the moment as well. So it's my absolute pleasure to introduce Brian, and he wants this to be an interactive workshop. So please ask questions as you're going along, or make comments as he's going along. Thanks, Brian.
Thank you, Belinda. And I'll reiterate, please, that, well, first of all, I am not an expert on microplastics. I've only been at this for a short time. So there's probably some of you in the room who have vastly more experience with this. So I'm not going to lie to you. I may make a mistake, but so I please, if you have questions or comments along the way, I want this to be an interactive experience. And it's kind of a history of the lessons that I've learned as I've dove into this process over the last couple of years. So let's move forward. So the reason I'm actually down here on the opposite side of the world is that I was fortunate enough to receive a grant from the Fulbright Association in the United States to work on a project that I entitled Penguins and Microplastics, a global collaboration on research techniques and population education. And the idea of this was to work with other penguin biologists who were just getting involved in microplastic research in order for us to try to coordinate and work together to do our practices in similar ways. So when it got around to analyzing those data and presenting those data, perhaps it would be a little more coherent instead of my lab doing it one way, Belinda's lab doing it another way, and colleagues in another place doing it another way, which, as we know, makes analysis of data a little bit more difficult. So I've been working, as Belinda said, with penguins in Argentina since the mid-90s. And did some initial collection of samples from penguins in Argentina in 20 and 21, and actually just went back. I was there in December this year and collected some samples. And the grant, the Fulbright grant, is working with collaborators in Chile. So I spent October and November in Chile, working with scientists at 2 universities there. Next week, I'm off to New Zealand for 2 months to be working with collaborators at the University of Otago. And then in June and July of next year, I'll be going to South Africa to work with some colleagues there. All of us, again, intended to work together. Some of these locations have very little experience with microplastics. Other locations, and I think here included, have scientists that have done more microplastic research. So trying just to get us all kind of collaborated and working together. And since I was in the neighborhood, coming to New Zealand, Belinda invited me to pop over, which, so I popped over. So here I am in Perth. I've always wanted to come to Perth. I was a map boy when I was little, and sidebar. I always get off on sidebars. If you take a stake and put it through the world I live in Bridgeport, Connecticut, it comes out about 200 miles off the coast of Perth. So for me, this is literally the other side of the world. So I've always been excited to come here. And I was fortunate enough to go with Belinda and Monica yesterday to see some of the stuff that they've already started doing with microplastics. And it was just a great experience interacting with both of them and talking about best practices and ideas and things like that. And, I learned some things from Monica and the work that she's doing, and perhaps she learned some things from me when we were out there yesterday. So I'm just excited to be here. So probably most of you in the room these are going to be no-brainer slides, but I just put them up there to make sure we're all on the same page. What are microplastics? So microplastics, by definition of that term, are small pieces of plastic that are less than 5 millimeters in length. kind of a broad definition. We classify microplastics as from two different locations. Primary microplastics are those that were actually created, manufactured small bits of plastic, typically used in things like cosmetics or soap, things like that. Abrasions, oftentimes some paints have microplastics in them. I'm not sure about what happened in Australia, but in the United States and many other locations, these have been outlawed, or they've been they're not really used anymore. I'm not sure if are they. Does anyone know if they're still being used here in Australia? They might be. They are, but they tend to be the prevalence of that classification of microplastics seems to be decreasing because we're recognizing the problems with those. The classification that's much more apparent to all of us are secondary microplastics, and these are the ones that come from breakdown of larger plastic particles, and so these are the ones that are more aware and certainly are not decreasing in prevalence in the world today. Where are they? We know now that they're everywhere. In particular, they're in general, they're really focused in water systems because they wash down from wherever else. So we find them in oceans, we find them in freshwater ponds, we find them in our drinking water. We find them in the air and the atmosphere. There's probably microplastic particles, unless you have an excellent filtration system, purification system here in this room, they're probably around us here. Find them in the ground. We find them in our food. Numerous studies have taken samples of hamburger meat, for example, and have processed the hamburger meat from McDonald's in the United States or Burger King, and we found there's microplastics in those hamburgers that we're eating. As we said, man-made products, cosmetics. One of the most prevalent locations to find microplastics is when we wash our synthetic clothes. So a North Face jacket, you wash that in the washer, and upwards thousands, if not millions of microplastic particles can come off of those every time we do the wash. So, and I put organisms with a dot on there because we now know that they're found inside of us, inside of humans. This is just some slightly older data showing you how the importance or the focus on microplastics in the literature has been increasing. In the early 2000s, there was literally almost no articles or publications that were out there about microplastics. And you can see over the last 20 years how the prevalence of studies in those different foci area have increased. And biology means found in different types of organisms. This shows the taxonomic distribution of the studies that have looked at microplastics in organisms, and you can see that the majority of them are obviously in aquatic species. For those of us that are interested in the most important group of animals in the world, the birds, you can see that there's a relatively low percentage of studies in birds. And the field versus laboratory obviously means they're collecting data from the field, and then these are more experimental studies in the laboratory. So my motivation for moving from stress to microplastics, I've justified it to myself and my colleagues in that we're still talking about anthropogenic effects. I spent, as Belinda mentioned, upwards of 25 years looking at the effects of tourism on Magellanic penguins and other penguins, primarily in Argentina. Magellanic penguins are colonial nesting birds, and in Argentina, one of the main colonies is Punta Tombo, and it's a very important economic industry there. And there's over 150,000 people a year that visit this penguin colony. I think Phillip Island in Australia is the only other colony of penguins in the world that has a higher number of tourists. So, and you can see how close these individuals can get to the penguins in Argentina. So when I first started working there, this was of interest to me. And we could talk for probably much longer about the stress physiology stuff that I've done over the last few years. But, I could tell you that no matter what colony that you look at, whether you're a colony that has had, does this work? Whether a colony that you've had lots of tourists for lots of years, or a colony that was just recently open to tourism, the birds that live in tourist areas are much more habituated to having those people around. And this is a measure of stress hormone, glucocorticoids, corticosterone in birds. we have cortisol. And doesn't matter if you have lots of penguins, lots of tourists or not, these birds habituate. We could talk about when you capture and hold a penguin at a colony that has lots of tourism. For whatever reason, if you've had tourists around, your stress response in response to a very severe event is actually lower. And we know that there's actually a physiological modification in the adrenal glands that cause this difference. We could also look at some really interesting physiology in how young birds are affected by tourism. Normally, animals that are born very, very young have very low stress responses when they're first born or hatched. And as they age, that develops rapidly. But we know in tourist areas that are highly disturbed, young chicks have an adult-like stress response 5 days after being hatched. And so how that happens and what happens with that has been the focus of a lot of my study. But we'll move on. Why switch to microplastics? A number of reasons. First of all, as Belinda hinted to, this is a topic that's getting much more prevalence and importance in the literature. So the studies of these have potentially broader impact and let's just say it's a little sexier than stress responses. This work is much easier for student participation. My university is a strictly undergraduate institution, so I don't have any graduate students, master's or PhD students. So it's much easier for my students to design and implement studies looking at microplastics as opposed to physiological studies where we're capturing animals and taking blood samples and measuring hormones. So it's an easier, a better topic for my students to work on. It's less invasive for the animals. Catching a penguin to take a blood sample is pretty stressful and pretty invasive. Typically with microplastics, you can just walk up and, like yesterday, we saw a lot of little blue penguins, but picking up the fecal samples was not any more disruptive than when Belinda was checking the nests. So it's a better way of, it's a less invasive way of doing this. It's less expensive. I don't need to travel to Argentina to collect and do microplastic studies, and the actual analysis of the samples once I get them is less expensive in the lab. And again, as I just mentioned, it's easier for my students to study stuff closer to home, so we don't have to travel that far. So in my twilight years of reading, maybe I shouldn't say twilight years, but I've decided to do this focus on microplastics, but I wanted to segue and keep my tie to penguins to start. So the first set of studies that we did, we're working still in Argentina with the scientific research. They're probably much like CSIRO here. CONACESIMAR is the Argentine research group. And then 2 aquariums close to home in Connecticut. So this is the East Coast, the New England area. Here's New York City, where most of you are probably familiar with. Fairfield University is located just about an hour east of New York City. And the 2 aquariums that we worked at were in the Central Park Zoo right in Manhattan, and then an aquarium just off the coast in Connecticut called Mystic Aquarium. In addition to that, we collected samples, initial samples from 3 different colonies back in Argentina. Punta Tombo, the colony where I've worked at for 25 years, we collected samples in 2021. And then, as I said, we just returned in December from collecting samples from 2 other colonies and also a second set of samples from Argentina. So this was our initial kind of data collection to start looking at microplastics. But our project was really to set up a laboratory, an analytical laboratory in Connecticut, to try to troubleshoot and develop best practices for isolation and identification of microplastics. And that's really what I want to talk about today, is this process of the ups and lots of downs of setting up that process in my laboratory. So again, we had the penguins from these different locations. I'm not going to show you. I have some at the end if we have time. I'm not going to show you any of our data yet, because a lot of it is actually still preliminary. But instead, I want to talk about kind of this process and how we've done it. I'm not saying that this is the only way to do it. I'm guessing that some of you in the room have done things in different ways. And I would like to encourage us all to talk about this and just think about, as we move forward, some of the things that I think are really important and some things that I've discovered along the way that were totally surprising to me. So this is just the penguin spread of the different samples that we have. So we have from the aquarium, we have Mystic, we have African penguins, 4 different species in Central Park, 2 species in the wild. In Chile, when I was just there, we had hoped to collect samples from Humboldt penguins, but El Nino kind of messed up our studies. And with El Nino, the Humboldt penguins didn't come back this year. So unfortunately, we don't have data from Humboldt penguins. New Zealand will be adding little blue penguins and yellow-eyed penguins. And then, of course, in South Africa, we'll be adding South African penguins. I have to give credit where credit is due. None of this work would have gotten as far as it would have gotten without my students. And these are 2 undergraduate students who have been absolutely instrumental in helping me get all of this work to where it was. Gabe Rodriguez worked with me for 3 years. He went to the dark side. He's now in medical school, but did a lot of the grunt work of troubleshooting a lot of the techniques. And Jada Ormsby, she's a senior this year. She just was with me in Argentina collecting samples in the field, and she's been doing a lot of the more advanced studies using some of the interesting microscopes that we've got, which I'll talk about in a little bit. And this is at Mystic Aquarium in Connecticut. So again, what I want to talk about now and interact with you and listen to your suggestions or your similar pros and cons about how you've done this are best practices for field work and laboratory analysis of microplastics. The first thing I think we need to think about is what samples we want to collect and what species we want to collect these from, all right? Why do we want to think, I think why is it important to kind of think about this beforehand? This process is time consuming. All right. Yep, it's easy to go pick up a fecal sample, but from there to identifying what those plastics are is a time-consuming process. There are cost considerations. Yes, it's cheaper than glucose physiology studies, but it can still be quite expensive to take these samples all the way to identification. The accuracy of results, I think, is one of the most important considerations that we have to think about. And I'll hit this now, but I'll come back to it. Contamination is a huge, huge issue with microplastic studies. I told you, they're everywhere, right? And if we're not very, very careful about trying to reduce the potential contamination from the air, from the vessel that you study, that you put it in, from the fluids that you use to treat the sample, so on and so forth, I could tell you that there's PVC or other polystyrene or things like that in that sample, but if you didn't collect that sample and protect that sample from contamination along the way, we really don't know whether that plastic is really from that fecal sample. And I forgot to actually put the reference up there, but there was a recent study that hypothesized or that suggested that upwards of 70% of the published papers on microplastics that have been out there are probably contaminated. 70%, that's a lot, all right? The other thing what you want to think about is the relevance of these studies. I just got an email from a friend of mine. Oh, Brian, I was sitting in my backyard and a robin pooped on my picnic table. I picked up poop for you because I know that you look for poop, you look for microplastics in poop. So I collected a sample for you, all right? Great. A robin in America probably does have microplastics in it, but am I going to spend the time? So I'm just, the relevance of the group of organisms that you're looking at. Is it a species that might be particularly affected for health reasons? Is it a species of conservation concern? American robins, they're doing just fine. All right, so these are the questions that we need to think about as we design our studies and move forward. Problems of definition. Microplastics, I said before, are strictly are pretty consistently defined as any plastic particle less than 5 millimeters. Okay, well and good, but there's plastic particles that are bigger and there's plastic particles that are smaller. And some people might argue that macroplastics, right? In fact, a number of people have commented to me that, you know what, and I put this up here, right, I'm an American, and we use the different systems. So I have to have this up here so I remember the conversion between all the different metric units. So I was supposed to take it out, but I forgot to take it out. Macroplastics, some people will argue, is what we really should be thinking about, all right? There would not be microplastics if we didn't have macroplastics, all right? I have an economist friend at my university who I'm actually doing some collaborative studies with, and he sat me down, he said, Brian, what you're doing is interesting, I understand with the penguins and everything, but at the end of the day, how are you going to solve the problem of microplastics until you solve the problem of macroplastics? So really, should this be what we're focusing on? Should the money that we're suggesting to spend on microplastic studies instead be focused on macroplastic studies? And in fact, you know, as I talked about it with my colleague, we see pictures of these large rafts of plastics in the ocean. And perhaps for you folks on the Pacific Coast closer, it's a little more relevant for you. But for someone from New York City or from Minnesota in the middle of North America, the impact of this on their lives isn't really all that important, all right? Large rafts of plastic in the Pacific Ocean aren't really affecting my life directly, all right? But what we now know is that there is definite evidence that these plastic particles are affecting health in humans and in other organisms, all right? So my argument back to him was, well, but look, all right? People are documenting that these plastic particles are actually affecting the life of organisms. So yep, not the Pacific, but they are having effects on you. So we need to figure out this bigger picture because ultimately it's affecting our lives as humans. Well, my economist friend said, but Brian, those are nanoplastics, okay? Plastic particles that are less than 5 millimeters and bigger than 1 or 0.5 millimeters, those aren't passing across the intestinal barrier, right? What you're seeing in bloodstreams, in tissues, and things like that are nanoplastics. So then we shouldn't be looking at microplastics, we should be looking at nanoplastics instead. And most of the papers that we see talking about nanoplastic or microplastics are probably incorrectly making assumptions about the particles. For example, I was just at the International Penguin Conference. Belinda was there. Lovely set of researchers had their poster next to me. This lovely student had a beautiful photo of a 3 millimeter fiber from a kidney sample in the penguins that she was studying. And she was very excited to say, look, I've documented microplastics in the kidney of these penguin. And I didn't have the heart to tell her that that is absolutely not possible. There's no way that a 3 millimeter fiber is going to pass across the intestinal barrier and end up in her kidney. It was probably from the air in the room, perhaps the clothing she was wearing, so on and so forth. So these are just some of the issues and problems that we're looking at. And this is just a slide I put up here to show you that there is extreme documentation that small plastic particles, nanoplastics, are present in tissue. So this is, what was this, from different sizes of plastic particles along this, and then I don't remember, brain, epididymis, I can't quite see the tissues in the other ones, but showing that there is indeed evidence of plastic particles, microplastic particles in tissues. So it is affecting us. One thing that there is growing evidence of is that perhaps it's not the actual microplastic particles themselves that are the issue. It is that they are vectors or vessels for bringing in other toxicants. So toxic chemicals, viruses, other things like that, hop onto those microplastics, get into the digestional system, and are a way of delivering those things that cause negative problems. So it's not the plastics themselves, but that they are useful in moving things forward. And this is just a couple of cartoons showing you the results of some studies that have documented or have tested in the laboratory the ability for plastic particles to bring in things into bodies and that can cause negative effects. One more, just talking about some of the evidence of physiological damage that has been attributed to microplastics bringing in negative aspects. So let's talk a little bit now about problems of methodologies and techniques. Okay, and again, I'm going to step back and say, what I'm starting to do is what I've developed in my laboratory. And this picture shows you that there's a lot of variation in techniques and methods to look at microplastics. This is just a set of studies from as distant as these are mussels here, marine mammals, birds, whales, reptiles, other things, showing you the variability in techniques that different laboratories have used in order to determine what's coming out on the other end. All right. Just so happens that over the 2 years of work in my lab, this is what I have determined are the best techniques for me. I'm not here to tell you that this is what you should be doing in your laboratory here, but this is what has worked out for me. And we're going to kind of walk through the main different components of collecting and analyzing microplastics and discuss where some of this variability can come in. And some of it's going to be intuitively obvious to you, and some of it may not. So these are kind of the different sublevels of processes along the way, from collecting your sample in the field to ultimately identifying if there's microplastics in there and what they are. All right, so let's start with, and remember, it's contamination at every step along the way that you just need to keep in mind. I think if a biologist walked in here and said they have developed a microplastic analysis protocol in their lab that is 100% contamination free, I would take that bet and guess probably not. All right, so at the end of the day, you're probably gonna have to deal with contamination, but the point is keep that in mind and try to figure out how to correct for that or admit that this was a contamination problem. So collecting samples, okay? In the field, something simple as what to wear, right? If you're a field biologist, especially if you're working like in cold temperatures, most field gear that protects you from the cold is made up of synthetic materials. And as you're out there running around in the cold, those synthetic materials are shedding things. So actually, the best thing to do if you're a field biologist and doing microplastic studies is to do it naked. You're supposed to laugh at that. Because then you won't be having any microplastic, you won't have any plastic particles on you. So we try very hard to wear to wear natural fibers, right? Cotton fibers, which will ultimately be digested in the process along the way. In Chile, on the islands we worked at, it rained really, really bad. I don't have, and as you can imagine, cotton rain gear isn't all that effective. So I had to wear a synthetic rain jacket. What will I be doing when I analyze those samples? I will also analyze a piece of that rain gear to see what kind of plastic it is and to see if that plastic spectra appears in any of my samples, all right? I wore the same rain jacket all the time. I didn't have other people working on the samples, but there's nothing else I can do, all right? So I just kept that in mind. This just shows you cotton gloves. So oftentimes for other reasons, in Chile for health reasons with the avian influenza virus that was around, we were instructed that we had to wear latex gloves or nitrile gloves to protect us. So we put cotton gloves over the top of those to help potentially block any fibers from that. Tools that we collect with, all right? Can't use a plastic, shouldn't use a plastic spoon, all right? So we use either metal tools to collect samples off the ground, or recently we've also started using tongue depressors. You know, your doctor, wooden, break in half, can easily scoop up samples, and the best thing about them is you're done and you can throw them away. These implements need to be as clean as possible. So before I go to the field, I rinse all of my metal spatulas in doubly filtered water to remove microplastics from the water, wrap them in tin foil, and off I go. All right, you clean your tools, and then you throw them in your field bag. Your field bag probably has synthetic material in it. So now you're just back to almost to square one. For me, I try really hard to collect samples that are fresh, i.e. I'm out in the field, and normally, and it's very much depends on the species, I won't collect a sample unless I see the bird produce that sample. Then I know that it is relatively, had a very short time of exposure to plastic contamination. Little blue penguins that we worked with yesterday, if we had to wait to find a little blue penguin poop for us, we would still be there 24 hours later and probably not have more than 2 samples. So for little blue penguins, we collected relatively fresh samples that were on the substrate. All right, so you have to also think about the biology of your animal. All right, and we take that in mind. What we put them in, we scrape up the fecal samples and we put them into tin foil. Both Monica and I use tin foil. I was just shipped a bunch of samples from a very, very prominent Antarctic biologist who has fecal samples for the last 10 or 15 years from a bunch of different penguin species. She's got a lot of money. She said, I'm sending you these fecal samples, please analyze them. They arrived in lovely little plastic bags. And when I asked her how she processed those samples in the Antarctic, she said, well, we just went out and we collected them, and you know, I've worked in the Antarctic and I know what they wear. So there's probably potential of contamination from her clothing. And then she showed me how they dried the samples, and her very, very talented graduate student was in a hut in the Antarctic wearing his North Face jacket, and little piles of poop were on the desk and he was drying them, all right? All of those things were potential locations for contamination. And so she and I talked about it, and I said, you know, at the end of the day, I'll take those samples that you got me, and I'll tell you if there's plastics in them and what they are. But are you really going to know that from that penguin on that island, or was it from what you were wearing? Was it from all these other things? So in spite of all the money that she had, we basically decided the time and the ability to really know for sure if these are microplastics wasn't worth it. And it's unfortunate. 10 years, probably 1,000 different samples that would be really interesting to see differences in microplastics, but it's just not, unfortunately, worth it. Storage, proper storage, I just mentioned. She sent me these samples in plastic bags. Now, to her benefit, they were the same type of plastic bag. She only used whatever it was. So we could take a sample of that plastic bag, analyze it, and subtract or correct for that if possible. But again, that was just one of a myriad of issues. So that's just getting the samples from the field, all right? This just shows you some of the variability of samples as well. These are the samples from the 4 different species in Central Park Zoo. So one would think that they're very relatively consistent, but size, other situations can vary in how you're doing your analysis. One that was really interesting, and I should have realized this, we did a lot of the troubleshooting of these studies with these captive birds, and in aquaria, birds pop out onto a nice cement substrate, poop, and the curators go and collect it for us. So that's a aquarium penguin species. This is a penguin. This is the sample from a Magellanic penguin, adult, that pooped on the ground. Right? I don't handle the adults. Okay, so by default, those samples have lots of soil in them, and that affects how I'm going to do the analysis in the laboratory. So all of these things you need to consider. I may have the best microplastic lab set up, but if you send me a sample from another organism in another kind of scenario, I'm going to need to start from scratch and figure out the best way to analyze that sample from where it comes from and what you've done. All right, so after we've got the samples, all right, we have to figure out how to process them. And the two main things that we want to do when we have a microplastic sample is get rid of any organic material that is in there and try really hard to float any of the plastic particles that are there. All right, so one thing I'll mention before we move on, I think it's important, and it's often not done, if you have the ability to do it, to do some ground truthing of the effectiveness of how you do this in the lab. So for our first 4 months, every sample that we processed from the zoos and aquarium, we spiked with little orange plastic balls. We put 10 little tiny plastic balls into every sample that we processed, all right? And we put it through our entire process, and at the end, and I'll show you an end product, we had a filter and we saw how many of those little orange balls were there, all right? And I can tell you, very rarely were there 10 little orange balls at the end of the process. What was hard to do in this process, because there were orange balls, is as you're doing these things, oh, I left an orange ball there, so I'm gonna go back and find it. And you gotta let that go. Because when you're studying microplastics, you're not gonna see a little plastic fibers that's there. All right, but I think it's important for you to kind of determine how good your lab is. All right. Talking about digestion, as I showed you on that cartoon of all the different studies, there's lots of different chemicals, and I'm not a chemist by any means. And so if some of you are chemists, you probably have much more experience or could offer me much more recommendations about what might be the best thing to use. In most of the studies that do this work, the two primary chemicals they've used for organic digestion are hydrogen peroxide and potassium hydroxide. All right, we started with potassium hydroxide. Tends to be a little cheaper and a little easier to get, but potassium hydroxide is very caustic on metal. And Gabe, the student that I told you, my undergrad, God love him, he was a bit clumsy. So he spilled and he splashed a lot. And in our laminar flow hood, after his summer of research, the bottom panel was so deteriorated by his splashing that I had to pay $1,000 to replace it. So we thought, you know, maybe that's not the best thing to use in a laboratory where undergraduates are doing most of the work. So we switched to hydrogen peroxide. It's a little bit cheaper. It doesn't have that cost effect on the metal, and also it's less problematic when it gets on your skin. If it gets on your skin, it burns a little bit, but it's not horrible. So we chose to use hydrogen peroxide, and we typically start with hydrogen peroxide that's at 30%. Right? You can't buy, well, maybe you can buy it here, but I doubt you can buy over-the-counter hydrogen peroxide in Australia from the pharmacy. Usually at most it's 10%. In Chile, I could only get 3%. So we start with laboratory grade hydrogen peroxide that's 30%, and then test to see what volume is useful for properly digesting, and or primarily volume. Two other things that are useful in helping breaking down organic material is heating the solvent up and mixing the solvent. I don't think I put it, so this is a picture of some of our samples that are in the digestion process on a mixer, and what you don't see very well is that is in a oven, all right? So I put it in there overnight. I typically start, when I get a new set of samples, at 30% hydrogen peroxide, 4 times the amount of hydrogen peroxide as the volume of the sample that's in there. In the sample overnight, shaking at 100 RPMs and temperature about 45 degrees C, which totally throws my students off because they have no idea what 45 degrees C is, okay? Oftentimes that works well, sometimes it doesn't. Again, if you're a chemist in the room, you probably know that if you have a substrate that's a high organic content and you put 30% hydrogen peroxide in it, heat it up and shake it, it turns into a little volcano. So there were numerous times when I came back the next morning and they didn't look like that. They looked, I should have found some pictures. There was a disastrous mess all over the place. So what do? We go back, we cut the amount of hydrogen peroxide, or perhaps we change the volume, so on and so forth. So these are all kind of the steps in figuring out the best way to get rid of that organic material. After you've attempted to get rid of most of the organic material, again, I mentioned that density separation is a useful step. It's not always used, but if you add a liquid that has higher density, obviously, it increases the density of the fluid and more things will float up. The literature that I've looked at, the best solution to use is sodium iodide. Costs a fortune, right? So we choose to use sodium chloride, so salt, supersaturated salt solution that we put in equal amounts with the hydrogen peroxide that were there, float things up, right? Is it the best? No, but it's the cheapest and most practical for what we are doing in our lab. Yes, ma'am. Yep. So the covers are there to try to prevent any environmental contamination from the air. So that's the first thing. And why they are, I can never remember which is which, why the bulgy part is down. As they're shaking, that allows them to, it literally is kind of a physical, if they're upside down, they can fall off much easier when they're shaking. All right, and if anything splashes up onto that, it will tend to drop down rather than drop to the side. Yep. You could use tinfoil. If you were using tinfoil with the potassium hydroxide, the tinfoil, if it got splashed on, and we wash this and we reuse it. Yep. So our tinfoil, we do not use, and Monica and I talked about this yesterday, the easiest and the cheapest thing to do would be to get some tinfoil from the grocery store and cut it and make it into sizes. A couple of issues with that. As I'm cutting it and down to size, I'm cutting it in a room that's open, and so we use laboratory tinfoil that's already cut into squares, and it comes out like Kleenex. I don't know if you're familiar with that. It looks like a Kleenex box, and you just pull a piece of tinfoil out. And we've tested numerous times that so as you pull it out, it stays bent in half. And so we've tested the inside of that tinfoil and have found no evidence of microplastics in there. So that's what we use. It's more expensive, but as far as time and contamination is concerned, it's a little bit easier. Remind me as we go further, there's also even more expensive. You can get tinfoil that's already cut into little squares, and we use that for covering dishware after we've washed it and put it to the process again, so we're not cutting things down. Even the big sheets of tinfoil from laboratory tinfoil, cutting it down adds a process. One other thing, oftentimes tinfoil has a coating on it to, I don't even know why, but so you need to be careful to make sure that the tinfoil that you get, even laboratory tinfoil, often at least one of the sides has a plastic coating on it. So you need to be sure that the tinfoil that you're getting doesn't have that. The little squares of tinfoil that I mentioned come with a piece of something when I got them in the middle, so it's easy to pull them apart. I had to call the company and say, okay, please tell me what is that piece of something made out of? And they assured me that it's regular paper and there's not plastic. I analyzed it and that was true. So all of those things are things you would never think of. It's tinfoil, right? It's only aluminium. That's how you say it here, right? So all of these things, I mean, and these are good questions to ask because these are things you don't really think about. It's tinfoil. Filtration system. All right. What I don't show you well is that this is being done in a laminar flow hood. All right. And there are many, yes, sir. Yes.
So presuming that is all types of plastic, that is different types of microplastics could be there. So did you check like whether this percentage degrade the plastic into micro or nano form? There is a chance.
There is, you're absolutely right. There is a chance that the solvent that you use will affect the plastic that's there. Yeah. That's just something that we can't control for. And we're hoping that the degradation is such that it will still appear as a plastic, even if it's a degraded plastic when we actually get to the identification process. But you're right. One other thing I was going to say about the percent of the peroxide, I don't remember what I was going to say. But yes, I mean, the chemist in the room will be much better. And also the flotation with the sodium chloride, there's just certain plastics that won't float in supersaturated sodium chloride. So they're going to be left behind. If I was using sodium iodide, there would be a higher probability they'd be floating, but I can't afford to use it. Right. So all of those things are considerations that have to be made when at the end of the day you're reporting your data. So this is a classic Buchner funnel filter system. This gives you an example here showing you how all of the glassware that we use is washed and is covered in tin foil until we're ready to use it. One thing that we discovered 3 months into this process, and I didn't think about it, we have a very nice acid washing dishwasher in our university. So we were putting our glassware in there, and as soon as it came out of the dishwasher, we covered in tin foil, off we go. As we got into it, we were starting to think more about looking where sources of contamination were. So we took water from the dishwasher at different stages in the washing process, filtered it, and looked for microplastics. There were microplastics in the water from our super expensive dishwasher. So after we've washed it, every piece that comes out, we hand rinse in double filtered water. So my students spend probably half of the time in the lab washing dishes. All right. Wouldn't have thought that that fancy dishwasher would have microplastics in it, but when you think about it, all the tubing coming into it is plastic. All right. So that just shows you an example of how we have to keep it. Filter system, classic Buchner funnel system, you can see that they're ready to go. Each sample gets a different funnel on top to keep the different samples situated. Yes, ma'am. Yep. Yep. So every, when we make our sodium chloride, we double filter it. When we get the hydrogen peroxide from the distributor, it comes in a plastic jug. We double filter it, keep it in glass bottles. All of the water that we use for making the salt, so on and so forth, we double filter it. So everything is double filtered. Probably overkill double filtering it, but we do double filter everything. Yeah. So this just shows you then the examples of filters that have resulted from filtering the actual sample. And this one you can actually little orange balls that were left behind. So that is 20% efficiency right there. This one also 20. There's 1, 2 there. This one's a good one. 1, 2, 3, 4, 5, 6, 7, maybe 8. All right. So that just shows you, and this kind of gets to the point, and also, sir, to your point about how the chemicals affect the process. I would be very suspect, and I would not present my data at this point, given how we analyze things, to say that this sample had 8 fibers and that sample had 2. And so this penguin was more exposed to microplastics than that penguin was, because of variability such as how the solvent affects the plastic fiber, how many are left behind, right? I just don't think we're there yet to make those kind of comparisons. Can I say there's microplastics in these samples? Yes, but I just still don't think we're at the point of being able to compare small things like males versus females or things like that. It's kind of a sidebar, but so then we take, and this just shows you, so after we get, oops, go backwards. After we get the samples put onto a filter, the next thing we do is we try to look for potential plastic fibers. And I say fibers, right? Because first of all, those are really easy to see, all right? And relatively easy to analyze. But the problem is that not all plastic bits are fibers. All right, so it can be very difficult if you're using a dissecting microscope like we're doing here, and we're finding these fibers to say, well, maybe this happens to be a bit of foil, but that might be a plastic bit as well. So our studies are focusing on potential microplastics in fiber form. All right, that's the limitation of, has been the limitation of our research capability so far. All right. We find those fibers. We, in our laboratory, use a dissecting scope. We photograph each of the potential fibers. We map out the filter and photograph each of the potential fibers so we can measure it using a measuring software. And so we can easily locate them when we take the filter to the chemistry lab to actually analyze what the fiber is. So we've taken pictures of notice this one? Looks like a penguin. I put it up there. Supposed to laugh at that too. So then in our early studies, we then tried to identify the fibers. And if you look at the literature, a couple of things are kind of the norm for previous studies. Many studies of microplastics presented their data based just on color. Right? So a lot of the initial studies said there were 5 blue fibers, 10 red fibers, so on and so forth. All right. Highly likely that those are anthropogenic plastics, but we don't know for sure. Also as well, most of the studies only take a subsample. If they do identify them to what type of plastic it is, they only take a subsample of the actual fibers. So if they saw 100 fibers, they'll only take 10 red ones, 10 blue ones, and take them to identification. We decided in our lab that we want to try to identify all the fibers that we see. And you can imagine it takes a long time, especially if you use an identification system that typically is available in most locations. So this is a FTIR spectrophotometer. We use it in our chemistry laboratories, so it's available upstairs in the chemistry department. And again, I'm not a chemist, but the take home here is if you take that fiber, which you've brought upstairs on your filter, and you know where it is, so then you find that fiber and you take it with a tweezer and you put it on the FTIR, all right? If you're lucky, if you don't sneeze, if there's not a gust of wind in the room, you'll transfer it over and you can generate an identity of what that plastic fiber is. I literally cannot do this, all right? My hands are too arthritic and tired. My eyes are horrible. If I didn't have my students to do this, I mean, I can find the fibers under the dissecting microscope, but there wasn't a chance in hell that I could do this. Right? We were losing, even with these guys, we were still losing 20% of our fibers to human error. Whether they lost it, moving it over, or it blew away, or for whatever reason, it wasn't there. The other problem with the system is once you put the fiber here and analyze it, it's gone, all right? One shot. Right? So it was the best technique that we had, but you can see the limitations of that. What happens, and again, this is layman's terms. You put the fiber onto the spectrophotometer and it gives you a spectra, okay? You take that spectra that you generate and you compare it to a library of known spectra of, and this is an important point, a library of known spectra, okay? Why this is an important point, the library matters, right? If most of these library systems, if you take your spectra of penguin-shaped fiber, put it on the spectrophotometer, compare it to a library that only has 10 spectra of known plastics, it will pick the best fit for that fiber out of the 10 spectra that are there. And you're probably looking at me going, duh, of course that makes sense, right? If the spectra has 10,000 things in it, it has 10,000 things to choose from. So when we first started, we were using a very, very large library. And this is not the best example because it's showing us that it's a microplastic, but we were getting things like sodium chloride coming up first. Well, of course, sodium chloride came up first because these fibers were soaking in sodium chloride. So we were having a very hard time. We wouldn't see anything until much farther down the line that might be a microplastic. So dumb field biologist me contacted the company and said, can you give me a library that's more specific to plastics? And they're like, here's your plastic library. It only had 100, all right? When we were using this library, we were getting about 60% at most, no, 40% of the fibers that we thought might be microplastics were microplastics, right, in the big library. When I switched to the microplastic only library, 100% of the fibers were coming out as microplastics. I'm like, oh, this is amazing. I thought, wait, something's wrong. But I didn't think about it. I wasn't smart enough. So I took one of my hairs, put it on the machine and analyzed it, and it came out as PVC. I'm like, that can't be right. And I called them and they're like, well, if you idiots, if you only give it 10 things to look at, it's going to pick the best fit. And it's going to pick that best fit at a pretty high probability or percentage. So this number tells you that it's 97% confident that it's PET. Well, if you only get 4 things to pick from, it's going to be 97% confident of those 4 things what it is, all right? If you look at the literature, many people do this and just pick the top one, right? If you talk to the chemists who really know what they're doing, they will tell you that if they look at this, they could tell you, oh, that's PV. They know everything. They know this little bit's that, and just looking at the spectra, they know. And so when I talked to the chemists when I was having this issue, they said, you have to look at the spectra. You have to dive in and analyze to make sure that what you're looking at is the same thing. My hair, the spectra looked nothing like PVC, all right? But because it said it was 97% confident PVC, I was like, oh, that was it, okay? Most undergraduates don't have, how do I put this the right way? It takes some training for you to be enable for you to look at a spectra and determine whether you're even close or not. And most of my students don't have that capability, right? So I talked to, well, let me jump ahead. So a great event happened in my laboratory is that I was fortunate enough to get a grant to purchase a brand new Shimadzu infrared spectrophotometer microscope. So now instead of having to look for the fibers and move them over one by one, I can take my filter, put it on right here, and using a computer can find those fibers and identify and mark them and identify them and get a reading for what they are. Right? It's great because we don't have to move them and they're not destroyed. In addition, although we're not quite, we haven't learned it yet, you can take the filter, put it on there, push a button, and it'll analyze the whole filter. So you don't have to find the fiber. All right, it'll give you a heat map showing you the different potential locations there. And if you know what to look for, right, you can go through and simply find all the fibers that are there. So this is potentially going to revolutionize the ability for our laboratory to identify fibers and to be very confident about them. All right. Unfortunately, that arrived and was finally set up the week before I went to Chile. So we haven't been able to really develop the tools yet. One thing, however, that we did work on with the company is that this microscope is super, 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 super capable. It can tell you the difference between a plastic fiber that was just off the press has been subjected to 5 minutes of UV contamination or has been subjected to 10 years of UV contamination, and it has the specificity to determine those differences. All right. I told them, I just need to know if it's a plastic. And the guy literally said, I don't know if we can do that. I mean, we had this conversation. I said, you just need to dumb it down. I don't know if you have the books down here, like Microsoft for Dummies and things like that. You need to be able to get this $100,000 microscope to be able to simply say, is this a plastic? Is this an anthropogenically derived structure? I said, and if you do that, these things will move off the shelf in no time. All right. So that also never dawned on me that something like this, it was like, you got to get this. It's going to benefit you so much. It has, but it's too good. The library that you use also as well. So all of these things are characteristics that I never thought about when I was setting up this microplastic lab that might be effective. So this just, this is to remind me to talk about the software analysis. What software you use, what library you use definitely matters. That's it. Oh, and just limiting contamination. So just reiterating once again, how you collect things, glassware. I have a second year student who just joined my lab, and his job, not very glamorous, is to take control samples once a week. So he puts a Buchner funnel system and runs it in the laminar flow hood for 2 hours and collects an air sample. He then takes it and he runs a Buchner funnel system in the middle of the lab, takes an air sample. He takes it to the room where we do the glassware, and he takes an air sample. He also every week samples our sodium chloride, our potassium hydroxide, or our hydrogen peroxide, our doubly distilled water. And I said to him, yeah, you're not out in the field, you're not collecting data, but this is the most important set of data that you're collecting. Because if you can't show me that the air that we're using is relatively, if not completely free of microplastics, or the chemicals and water that we're using aren't clear, then we got to stop. So that's his project. I mean, I can't reiterate enough the importance of limiting contamination. That's just showing me collecting a penguin sample from a Magellanic penguin that just pooped on the ground. So we have this hanging in my lab. Did you do your dishes? And right beside it, it says it's not science unless you do your dishes. So with that, that's all I have. Again, I'm not an expert. I'm sure that there are many of you who, or hopefully there are some of you out there who've had some experiences and offer some comments or concerns or criticisms. I can take it, but there you go. Yes, sir. Yeah, so not possible. That's another very important point. The way we were doing it in the lab now, if we're using the dissecting scope, all right, if I didn't have my new microscope, we couldn't. There's only so small we can go. All right, so that's a huge limitation. Not every lab, very few labs have a Shimadzu, $100,000 microscope. Most labs probably have access to the regular FTIR, but you can't get to nanoplastics if that's what you're using. Right? And if you can't get to nanoplastics, then you might argue, well, why do you do this? Because who knows what's still inside, right? So I have colleagues who are doing captive studies on model species. We have a study with tilapia, and then she's doing some sort of invert, I don't remember, where she's feeding them microplastics, plastic particles, letting them ingest, collecting fecal samples, but then we're also sacrificing the animals and seeing where those are going, right? I'll never be able to do that with penguins, and I'll never be able to feed microplastics to a penguin. We can in the aquarium, but we can only sample what comes out. They'll never let me collect a penguin and sacrifice it and see where those microplastics go. But perhaps with these captive studies, and we come up with some idea of the percentage of fiber, percentage of plastics that go in versus the ones that come out, we can then hypothesize, well, it looks like efficiency, so on and so forth. Yeah. I had started to do some stuff with eggs. So I think that, and that's easy to do because eggs are, and especially with the zoos, I don't know how they are here, but zoos in the US are notorious for not being willing to do anything remotely invasive with their animals. And I get it. I get it. When I was doing my stress physiology studies, I wanted to go to Mystic Aquarium and do some stress series on their young chicks. I've been doing it in the wild for 25 years, never injured a bird. They all survive. But there wasn't a chance in hell that they were going to let me take a blood sample at 0 and hold the bird for 30 minutes and take another blood sample. It's just not going to happen. I could have found, I think, some really interesting things about how stress develops in a captive situation, but not going to happen. That same zoo, the same aquarium, breeds their penguins, but most of them they don't let sit on the eggs. So they have boxes of eggs in their freezer that they're willing to let me have. All right, so here we now have a sample of eggs that that we're taking and opening them up and seeing if there's microplastics in there. We're starting with smaller ones because penguin eggs are big, but that's something my students can do, right? They can work with penguins, work with penguin eggs. So did I answer the question? I don't know where I went. I went off on a tangent somewhere. We're good? Yeah, it's interesting. So at my university right now, we are interviewing, the chemistry department is interviewing for a new chemist. And when I was in Chile in November, I got word from the chemistry department that one of the people that they're interviewing is a specialist in chemical analysis of the degradation of plastics. I'm like, oh, that'd be awesome. So I Zoomed her while she was there doing her interview process. And she said, oh, so tell me what you're doing. And I explained to her, and she's like, oh my God, you're doing it all wrong. I'm like, are you kidding me? You know, got this $100,000 microscope, and she's developed this technique using fluorescence only, and it's very simple and very straightforward. And she assured me that you can do this in your basement if you wanted to. So absolutely, yes, there are other techniques that are available to be able for you to determine whether they're plastics or not. And I don't understand them. I was pushed along this FTIR process, and if I told my benefactor that they gave me $100,000 for something that's not going to work, they'd be upset. I mean, it works, I think. Are there other ways to do it? Yeah, and that's another issue, for sure. I would say the answer to that is probably going to be no, because then I have this $100,000 rock in my laboratory. I also know that, you know, what she's done is probably very accurate, but also she's developed that, and that's new. For example, when I go to South Africa, they have just purchased, by coincidence, basically the same microscope that I have, different company, but same system. So that, in theory, with its cons or its pros, would potentially enable us to at least collect things in similar ways. We have a piece of paper next to it with a circle that's divided into 12 segments. And I put a dot on the top of the filter, cover it with a glass petri dish when it's under the microscope, because we don't do it in the hood, and literally scan back and forth. And whenever we find a fiber, we code it as being in X1, X2, X3, X4, X5, X6. And then when the camera is working well, we just take a picture of it. When the camera's not working, then we literally draw it out. So then when we take it, either when we used to take it upstairs to try to find it, or when we use the new microscope when we're not analyzing the whole thing, we put the filter on there and just use the microscope to get it and highlight it, and off we go. Yeah, I'm hoping that once I'm back full-time in Connecticut and can really dive into using the microscope, that we'll be able to use the tool where we put the whole filter on there and off you go. And there's a trade-off that takes longer, as you might expect. If you put a filter on there and just let it go and it does the whole thing, that takes longer than if you look at it first and tell the microscope where to look. If there's only one fiber in my sample, I think it probably is, well, and fibers, it's more time efficient to find it and tell the microscope where to go. The exciting thing about using the advanced screening is that we will be able to move towards non-fiber type things. And even down to starting to get into nanoplasticky ranges, which ultimately is what we want to do. Yeah. Are you Thomas?
Yes.
Okay, nice to meet you. We have, when we first started, we would take like every third and look at it before we put it in, and we were finding little to no fibers, microplastic fibers. To my fault, I have stopped doing that. I should probably do that more. But no, we don't know. Yeah, we use, that's another thing. There's probably better filters you can use, but they tend to be expensive the better you get. And some of these samples, like the fecal samples from Magellanic penguins in the wild, it's impossible to get all the dirt and soil out of there. So when we filter it, the filter gets pretty damn dirty. And so for one sample, I've had upwards of like 4 or 5 filters per sample. And if the filter is $5 a shot, that tends to add up more than $1 a shot. So those are other trade-offs. Yep. I mean, I have probably now, because we're backlogged, 1,500 to 1,800 filters ready to be put on the microscope. And if they were metal ones, I'd be broke. Yeah, and I mean, it was when I was given the money to buy a microscope and I was shopping around for a microscope. The two that I had narrowed down to was one by Brooker and one by Shimadzu, and I contacted both of them, and the Brooker folks were like, and I had already had filters ready to be analyzed, and like, nope, can't use them. You have to use metal, and I'm like, pardon my French, that's bad. And I went to Shimadzu and like, oh no, it's fine. It would be better, but you know, I use reflectance. I don't know, do you use transmission? Yeah, you know, and the Shimadzu folks were like, this is, you know, you'll be able to do this. So, you know, yeah. I mean, and that's the funny thing about these chemists. The Shimadzu folks were, you know, telling me how easy it is to identify these things, and you can be really, really specific, and you can get down to the nitty-gritty details of almost like how many minutes it's decayed. Simple to identify. And so I sent them a penguin poop sample, and I said, okay, find the fibers in there. And they're like, what the hell? I mean, this is not what we do. I mean, we work in these hermetically sealed labs and everything's perfect, and there's one little fiber on there, and you don't have to deal with organic material or the real world because it's all in this special room. And it's funny to watch their kind of like, wow. I mean, this is the classic difference between field biologists and laboratory biologists, right? They, you know, how many times have a geneticist say, send me some samples so I can identify, you know, this. You know, go to the field and get some penguin samples and send them to me. So, you know, it's interesting that these microscopes are so able to do what they can do, but give them like a real world sample that we want to do, and they just can't really do it very well. Yeah. Saturated salt? Yeah, you want to know how we do it? No. It's really technical. No, I'm worried that we're going to probably miss more than that. Yeah. And again, where I'm approaching this now, when I started, I was so excited to be able to, you know, compare differences between this colony and that colony and things like that. And the realization has hit me that in order to do that, I just am not really convinced that if you show me a paper that you say that you know, are finding those specific differences, I'm not convinced that you can do that yet. Yeah. No, I mean, I would love to be able to use sodium iodide, but can't. What are you using? Yeah. Yeah. Oh, yeah. Yeah. I mean, when I was in Chile at the end of last year, in my lab, we use, laboratory grade sodium chloride that we buy, and it's pretty expensive. And I couldn't get it there. So I just went to the grocery store and got salt and poured it in and let it spin until there was residual in the bottom, and off we went. And we double filtered it, and hopefully it's fine. Yeah. Yeah. I think the answer is that would have to be, of course. I don't want to I don't think that it's a good thing, right? So the short answer is yes, but I don't want people to say then this isn't a problem, because I think there are studies that, it's hard. I would say, and one good thing about some of these nanoplastic studies with tissues, if you're looking in embedded tissue that's been sectioned, I might argue that's probably a little bit harder to contaminate than something that's been exposed to the air. People might argue with me, but I mean, at the end of the day, really, we should probably be focusing all our money on macroplastics and getting rid of macroplastics, because that's where the problem is. Yep. Yeah, it's huge. I mean, I work with a woman in the U.S. There's a conglomeration of researchers in northern New England, in the northeast of the U.S. and across Canada, that have kind of gotten together, and we're working on this, basically a kind of a paper that has like almost a dichotomous key of what to do, where to go, so on and so forth. And at the end of the day, I mean, a lot of those lines end up with, it's not worth it. If you can't do this, it's really not worth it. If you can't do that, it's really not worth it, because it's just, and it may never be at a level that we can do those type of comparisons. Yeah, so you just made me sad and that I need to retire. I said, now you made me sad and I need to retire. Yes, sir. Go ahead. Yep.
And the reason they're ubiquitous is that they're so useful. And I would say it's probably one of the most useful products that humans have ever invented.
True.
But it's also one of the ugliest. And a point I've made to my wife is there wouldn't be any, she has a passion for growing plants in coloured plastic pots, that there wouldn't be an art gallery in the world with something on display made of plastic, because it's just too ugly. But it is useful.
Yeah.
And that's why it's everywhere. So it's interesting to see the questions or the solutions you've come to, and is collecting and eliminating all the other things.
Yeah.
I can understand plastics' physical effects, like the microplastics.
Mm-hmm.
Not talking about macro.
Mm-hmm.
Microplastics in blocking small vessels,
Mm-hmm.
anything from mollusks or vials. But the reason they're so popular is they're chemically neutral. Are there any chemical effects
Mm-hmm.
of microplastics?
I don't know the answer to that.
Probably another talk, maybe.
Say again?
That's probably another talk.
Yeah, and not given by me. I don't know the answer to that. I mean, as I mentioned, one of the growing foci of microplastics is them being a vector through biofilms and things like that for bringing viruses and other chemicals that are known issues for humans into the system easier. I don't know enough. I mean, I would, just as a dumb field biologist, I would hypothesize that, plastic bits inside of you can't be of benefit to you. I don't know. But I don't know of the studies that have, documented what the chemistry of plastics themselves, what, they can cause for human. Yeah. Yeah. Yep. Yeah. Yeah. Oh, there's lots of literature that shows when you have microplastics in the gut, how it affects the gut physiology. They're not passing across, but they're causing, inflammation or, other things that are physiologically changing the gut that have physiological effects, but they're not passing across. And so even, I could argue with my sister, yeah, they poop out, but there's still probably some in there that are having those, and there's some decent studies that have shown that, that just them being there are causing an effect to the, the biome that they're in.
So that's a correlation between corticosteroid and the plastic.
Well, that was interesting because that, when I was starting this, I thought that would be a cool thing for me to bring my two studies, my two foci together. I would have no confidence at being able to, I can measure the stress hormones in that bird, but if I collected a poop sample from that bird, I don't know if I'd feel confident enough to be able to, do a regression of how high your glucocorticoid levels are and how much poop, how many microplastics you have in your system. Yeah. I haven't seen them. Yeah. Well, I think I think you know what colleague I'm talking about from Antarctica who had those samples. She doesn't have from known individuals, but it might be interesting to see, in the fecal samples that she collected 10 years ago, if there was more incidence of plastics in there than the ones that or less than the ones she collected this year. When I was in Chile, it's an El Nino year there. So as you all probably know, El Nino means warm water off the western coast of South America, and warm water means no food fish. So the penguins didn't come back this year to breed. So I was there to try to find Humboldt penguins to collect feces from, and there were literally no penguins. They didn't die. They just are smart enough to know that when you can't feed your kids, you don't make kids. So on this island where we were supposed to be finding the fecal samples, the nests are in these cool grottos, these cool rock grottos, and it doesn't rain on the island. So there is guano buildup around the nests for years. So because I was trying to figure out something to do, what we did was we took initial scrapes from the guano, and I brought them back. And the idea is to see if we can find plastics in the upper layer, and if that's the case, try to figure out a way to do like a core sampling or to go down. It's going to be technically challenging, and one of the things with those is that guano is so concentrated with organic matter. I put 3 grams of it into 50 mils of 30% peroxide, and overnight it just went. So. And I didn't have time to perfect the laboratory technique. So when I go back, I'm going to have to play with it and figure out. But those are the kind of interesting questions that would be great. There is someone in Canada who's doing that with ice cores. And the benefit of ice cores is that A, you can core it and it doesn't pulverize, and B, just melt the ice away and there's plastic fibers there. So the techniques of potentially finding the fibers in ice is much easier because you're not dealing with nearly as much organic material. So they're starting, this one of the women I'm working with in Canada is doing those studies. I think those are fascinating, potentially. Or, who was I? Was I talking to you about this? Someone, maybe it was my colleagues in Hobart, about museum specimens. If there's a way that you could use museum specimens and look for microplastics in wherever. And I will say, sir, I went to the Mona when I was in Hobart, and there was some pretty dang interesting plastic art that was there. Any other questions? Thank you for your time. Thank you for the interaction. And if you could just turn the temperature down outside, that would be good.